Okay, so welcome back to the seminars actually. So we're very excited that we are expanding now to Solanaceae, thanks to the, also the help of it, that we are working this with Chelsea Pretz and Andres Orejuela. And today we are very excited that we will hear the talk of Sandra Knapp, that she's a merit researcher. You all know her, of the Plants Division of the Natural History Museum here in London. She is also the current president of the Linnean Society. She performed her PhD in the Cornell University, working on the genetic clade of Solanum. And after that, she continued working, as you know, in Solanum and also in Solanaceae, addressing multiple questions about taxonomy, nomenclature, evolution, and systematics in general. So, just to mention some numbers, she has more than 240 articles, like 25 books, and a lot of work also in outreach that we are very excited to hear. So now she will be presenting about the monographs and why monographs are such a big genus as Solanum, that she has been working with Tina Sarkinen, and she's also here. Tina is also a researcher in botany, but in the Royal Botanical Garden of Edinburgh. So we are very glad that you are all here to listen to this talk. And also, thank you very much, both Sandy, Tina, for presenting today. So now if you want, Sandy, you want to share your screen?
Okay, yay. Okay, so this talk is kind of based on a talk that Tina and I gave in a symposium about monography, about botanical monography, that happened in April last year at the Royal Botanic Gardens at Kew. So this is about a year old, but I thought it might be good just to kind of talk about why bother to do a monograph at all, but also why bother to do a monograph on something as huge as the genus Solanum. So this is a joint thing that Tina and I have done together. It's kind of our view of why bother, but the basis for this is everybody else. So everybody in the room has actually contributed to this in one way or another. So this is really our talk, all of us together. So now I just have to figure out how to make this go to the next slide. So when one thinks of botanical monographs, one thinks of books that have been published over the years that sit on our bookshelves. Many of us have these bookshelves. Chris Martin tweeted a really lovely picture of his copy of the Solanum in Australia monograph, and they become these reference books that we use for years and years and years. And oftentimes, monographs are not repeated for many years, and a monograph will last hundreds of years. Now, they're traditionally seen as being done by one person, and usually that's a man. There's one man in his office spending his life doing the monograph, and that monograph then becomes the gold standard for everything that comes after. But we no longer have the luxury of being able to spend an entire career doing a single monograph and for it to become something that is that reference forever after. The world today is a very rapidly changing place. This is a picture of the deforestation in Rondônia in Brazil in 2010. If I were to use an image from today, there would be even more deforestation in the Brazilian Amazon. And this is a picture which is repeated in many tropical areas all over the world. And you'll see why this is important in a minute. We also live in a time when we're looking very carefully at the limits of human exploitation of the world that we live on. And this is a figure which is from Rockstrom et al.'s paper from 2009 in Nature. They've done a different figure which says the same things, but I like this one better. And what this shows us is these are things which are human uses of the world we're on, and which of these things in which of these areas we've gone beyond the boundary of what the planet is able to sustain. And you can see that a very big one is climate change. We often think of that as being the biggest change, but actually the biggest and most uncertain change that we are effecting on our planet is that has to do with biodiversity loss. This doesn't mean that it's that has the greatest effect. This also incorporates uncertainty. So the uncertainty of what happens with biodiversity loss is what makes this such an important factor. Now, looking at the kinds of work that I in particular do, I want to kind of highlight two different things. And the first of these is this very smug picture of Carl Linnaeus with his trademark Linnea borealis. And in his Philosophia Botanica in 1751, he said, if you do not know the names of things, the knowledge of them is lost too. And this is an important concept, because if we name things, that allows us a hook upon which to kind of attach all the rest of the knowledge about that particular organism. Now, of course, names change, but we track those through time. And naming things allows us to talk about them. And I always say to classes when I'm teaching that imagine a world in which we didn't have names and we had to describe everyone in excruciating detail if we ever wanted to talk about them or tell someone else about something that someone else had done. It would make gossip absolutely impossible. In about the year 2000, people started to talk about the rate of taxonomic description, the rate of new species description. And the idea was that something had to happen to change this trajectory. You can see here that the trajectory, if we kept on describing species at the rate in which we were describing them in the year 2000, that the asymptote of this would be way out into infinity. And there would essentially be no end to describing life on Earth. Now, I'm not sure I believe in the concept of there being an end to describing life on Earth, and I'll talk about that a little bit later. So a big push was made to describe new things. And many of the new species that we describe are in fact not very well known. And this is partly because we're describing them very quickly. And this is an example of a new species that I described, well, Tina and Paul and I described this together in 2013. And we collected this in southern Peru where the black squares are. But then modeling the distribution of this using species distribution modeling, we found that it was probably also found in northern Peru, and in fact it was. So oftentimes the species that are described as critically endangered at the time of description actually are more widespread. It's just that we haven't used the tools that are available to us to actually look at how that distribution is on the face of the Earth. But it's important also to remember that a taxon name is a hypothesis about the distribution of variation in nature. So by naming something, we're creating a hypothesis that people can go away and test using new data, different kinds of data, different numbers of specimens, different DNA sequences, chemical data, all kinds of things can be brought to bear to test that hypothesis that this is something different from everything else. And Chris Martin and his group have been doing a really good job in Australia of looking at testing those hypotheses in an area where there were a bunch of species described, but then looking more carefully, those entities turn out to be quite different. It's also important to think about how the work that we do underpins work in conservation and work in understanding the planet and looking at how we can change our actions to conserve the planet better. And this is a figure which I've adapted from a paper that was done by Georgina Mace and her colleagues in 2014. And what this shows is the predictability, the vertical axis is the predictability from high predictability of something to look at how change is affecting an ecosystem to lower predictability. And this is how these persist through time, is about how these traits can be stable through time. And you can see that understanding of organisms, or the basis that comes from botanical monography, is actually really important, both at high predictability and low time in functional traits. What's a functional trait except a trait that an organism possesses? And also with lower predictability but more stability through time is that genetic library of life, of which the organisms that we study are the physical manifestation. So it's important to put our work in the context that it isn't an end in itself. It's actually something that contributes hugely to many other areas of science. So what is a monograph? The word monograph comes from the Latin, well, Greek, single writing. So a monograph is one place where the whole sum total of knowledge about a topic is presented. And the dictionary defines it as a single document that forms a complete text in itself. Or the Oxford English Dictionary, since we're here in England and it's the kind of last word on English anywhere, everywhere, is a monograph is a detailed written study of a single specialized subject or an aspect of it. Now, monography or monographs do not apply just to scientific topics. There are also monographs which are written about things like pottery. So these are Chelsea plates, Chelsea pottery plates, which are based on plants which were grown or illustrated by Philip Miller in the Chelsea Physic Garden. And there is an uncle of mine wrote a monograph on the Chelsea pottery. Now that's pretty straightforward because there are only about 100 of these, or less, or fewer, because several were destroyed in a house fire. There are only 100 of these, and these will never change. These are plates that were made in the 1700s, and there are very few of them, and they will never, ever change. Writing a monograph on something that is a moving target, the results of evolution by natural selection, is slightly trickier because that monograph is a moving target. So what about Solanum? Solanum is the love of my life. Solanum is something that I've spent many, many years working on, and I think everyone should love Solanum, and I'm sure all of you absolutely love Solanum. Now, Solanum is one of the top 10 genera in terms of angiosperm species diversity. So there are these 11 genera on this slide, from Astragalus with 3,270 species down to Croton with 1,223 species, comprise 6% of all of angiosperm species diversity. And the 17 genera with more than 1,000 species comprise 8% of all of angiosperm diversity. Now that's quite a large percentage in a small number of genera. And genera with 500 species or more are 16%. There are 57 of those, and those are 16%. So that means that quite a bit of angiosperm diversity is concentrated in these very large genera. So maybe those genera are large because people have already described all of the species in those genera. But thanks to Peter Moonlight, from whom we borrowed these data, you can see that these are some of those top genera. And you can see that the trajectory of species description between 2005 and 2015 is going up in all of these genera. All of these genera, there are species that are continuing to be described. And you can see Solanum, which we've outlined in green here, there are between 2005 and 2015, there were 118 new species of Solanum described. Now, this is nothing compared to the 341 new species of begonia which are described, but still the trajectory is up, which means that our knowledge of these large genera, the reason these genera are large is not because we know everything about them, but it's because they just are large. Now, Solanum has always been a big genus. It was one of Linnaeus's largest genera in his species Plantarum, and the number of species in Solanum just keeps going up. The biggest increase was between 1816 and 1852, when the results of the exploration of South America, which is the hotbed of Solanum and Solanaceae diversity, were coming to the European herbaria, where Michel Félix Dunal used them to describe all of Solanum in the last full monograph of Solanum, which was done in 1852. So Solanum's always been big. So maybe it's big because it grows in lots of places. Solanums grow all over the world in all kinds of habitats, from these kind of very wet forests on the eastern slopes of the Andes to the dry deserts of the western slopes of the Andes, to the high mountains in the Andes in Peru, to lower elevation forests, to southeastern Brazil. And they also grow in the arid interior of Australia, where a surprising number of species occur. And I'll talk a little bit more about that later. Now, they also encompass a wide range of body types. Solanums can be tiny little herbs. That's my thumb, which is holding this tiny member of the morelloid clade called Solanum Wedelii, to rather large trees. This is Solanum lycocarpon, which has fruits which are about the size of a baby's head. So maybe the reason that there are so many species of Solanum is that they have this broad range of body types and broad range of habitats in which they live. But to understand why there are so many, you actually need to understand how many there are, or what those species are. And in large genera, there are really big problems. And one of the biggest hurdles of all is synonymy. Now, a synonym is a species that was described by someone that a later person decides is the same thing as something else that was described earlier. So in Solanum, we have 6,168 names, of which 1,000, at the time that we made this slide, 1,234 of these are accepted, and 4,934 of them are synonyms. That means we have about an 80% synonymy rate. Now, in angiosperms at large, there's about a 33% synonymy rate. There are 3 synonyms for every accepted name. Looking at another, that's a lot. So Solanum has a very high synonymy rate. Looking at another big genus, Begonia, we have 3,987 names, of which I'm sure there's more now because Begonia people are describing species like there's no tomorrow. 1,925 of these are accepted. Only 1,089 of them are synonyms, which gives us a 40% synonymy rate. So actually, figuring out old names becomes a huge problem when you have a big genus and there's also a lot of synonymy, which is the case in Solanum. So Solanum hadn't been tackled since 1852 when Michel Félix Dunal wrote his great Prodromus. So in 2004, under the leadership of Lynn Bose and David Spooner and Michael Nee, we applied for a grant from the National Science Foundation in the Planetary Biodiversity Inventory Program, which was subtitled A Mission to an Almost Unknown Planet. And much to our amazement, we got this grant to do a global species-level taxonomic treatment of Solanum, and to make this treatment with all of the specimens and keys and illustrative material available on the internet. But also importantly, and this is where my perspective on doing botanical monographs has changed completely since before 2004, is to link this taxonomy of these wild species to emerging genomics data sets, which were largely done in cultivated species. So in 2005, I was invited to give, Paul Berry invited me to give a talk at the International Botanical Congress, which was held in Vienna in 2005. And so I put together a slide of the challenges in doing a monograph of something as large as Solanum at that time. And these are the challenges that I identified. First of all is time. Do we have time to do a monograph of all the approximately 1,500 species of Solanum? Do we have access to all of the material that we needed? Remember, this is 2005. This is before the Mellon Project and JSTOR Global Plants. This is before the broad-scale digitization of many of the herbaria around the world. Was there institutional commitment to doing this sort of work on a cycle where grants are every 3 years and institutions depend on people getting grants? Is there commitment from the institution to someone spending a long time doing botanical monography? There's a challenge with collective working. Remember the picture of Asa Gray I showed at the beginning? Monographs were usually seen as happening alone with one man and his microscope. Is there a way to give credit for collaborative effort on a website, for example? Are there common goals for something like a world monograph of a genus like Solanum? Can there be too many specimens? That's something that I've thought about a lot recently, and I'll come back to that in a minute. And also mission creep. It's very easy to get distracted into doing other things. So just keep those in mind, and I'll come back to those challenges a little bit later. So the first thing to do was to figure out what was Solanum. What was Solanum going to be? These are all the different genera that have been segregated from Solanum. And Lynn and her lab starting on with work from Dick Olmsted, did some phylogenetics and discovered that in fact, all of those segregate genera actually belonged in a 100% monophyletic genus Solanum. Now this was slightly controversial at the time because tomatoes were their own thing. They had their own community. They had their own horticultural community. It was important for people to think about what uniting things as a Solanum meant. And I just thought I'd put this slide in to think about how you divide genera up. How do you decide that something is a large genus or a smaller genus? It's largely a matter of opinion. We could cut Solanum here and have a monophyletic Solanum with 1,500 species, or we could cut Solanum here and have a series of groups, some of which were monophyletic, but whose relationships were very unclear, and then we would need to have a number of different names for all of these taxa. As a collective working on Solanum, we decided that Solanum, the broader concept of Solanum, was more robust, more phylogenetically supported, and last of all, it was actually something that was recognizable by people. Is looking at a Solanum flower, you can basically tell it's a Solanum from 5 paces. Even my kids could tell it was a Solanum. So we decided to treat this as Solanum in in the broad sense. And this turned out to be a very good decision because the monophyletic Solanum is incredibly robust phylogenetically through many different treatments phylogenetically. This is a treatment that Tina led on, doing a phylogenetic framework of the entire family using very, very broad sampling and also using dating, which Rocio is now looking to update. So looking at this in a phylogenetic context has been very important for structuring the work on how to do a monograph of something as large as Solanum. If we'd started with A and ended with Z, as did Michel Félix Dunal, we would still be on A. Whereas partitioning this phylogenetically allowed us to tackle such a big thing in a way that made it manageable. And the manageable way that that has occurred is through monographs of particular clades of things, or particular, sometimes monophyletic groups, sometimes geographically defined groups, but looking at things in manageable chunks that can be done in a smaller period of time so that we don't lose heart looking at Solanum. Now, many of these are published, and many of these are also published in the open access literature, which I'll come back to in a little while, because making things open access is really important for leveling the playing field and getting everybody up to a place where they can look at the information and use any information which is published. But we've also, and as part of our National Science Foundation project, also established a website, which is the website Solanaceae eSource, and this is an old screen grab of Solanaceae eSource. But what we have also done is groups that we have not monographed, but yet we've produced species descriptions of for other projects. We've put those directly onto the website without publishing them in the published literature. Now, we set up a mechanism of peer review of these descriptions, and those descriptions then immediately go onto the website themselves. So here's an example of one. We had a form that we would make the description in, so that the descriptions would all be parallel or more or less parallel, and then these would go onto the website together with pictures of the species concerned. So having those available on the website has meant that they're there. This is also a CC0 or CC BY website, which means that people are free to use that information if they cite the source. So that's also been important for subsequent floristic projects and a number of other things, that people don't have to start from zero. If they agree with our species concepts here on the website, they can use that description. The idea is for people to be able to use those descriptions to reformat, to be able to speed up taxonomic work considerably. So also a newer aspect of this is being undertaken by Rebecca Hilgenhoff, who's here in the corner. And what she's doing is doing that bit of the PBI Solanum promise that we never really did, is we never provided the identification keys for all of Solanum online on the internet. And so what she's doing, and this will be, some of you in the audience are testing this, and everybody will be able to test this and use it in a while, is to make open access, multi-access keys for which the descriptions, the ultimate descriptions, are then linked back to the descriptions and the information on Solanaceae eSource. So we're trying to make this into something that people can actually use. And we've done this as well by publishing keys to particular groups, particular continental areas, for monographs which were published not in the open access literature. So Maria Vorontsova and I published a monograph of the spiny solanums of Africa in Systematic Botany Monographs, which is not an open access journal, but we reformatted the keys, added new species to it, and made those keys available in the open access literature with, again, links back out to Solanaceae eSource where all the species descriptions are. So keys and the linking has been really important for us. Also important is the fact that, you know, there's a big wide world out there. And we certainly haven't collected all of the solanums in the world. We certainly haven't even seen all of the solanums in the world. So fieldwork has also been a really important part of this monography adventure with Solanum. We've done fieldwork in Africa, looking at species that we already know. This is Frank with Solanum anguivi. We've also found a lot of things that we don't know what they are. This is everybody pointing to something that's definitely a new species that doesn't have a name yet. And many members of the group have actually been describing new species. This is a particularly spectacular one, which was described by Yuri Govea last year, Solanum medusae, which is related to that wolf fruit that I showed you earlier. And here's one that's in press being described by Gloria Barbosa and I, which we found recently in central Argentina, in areas that are highly anthropogenically disturbed. And although this is a weedy species, this is probably one of the most endangered solanums that I've described recently. Even though it's found over a relatively wide area of distribution, its habitat is hugely altered. And you can imagine what happens when there are fields of peanuts for peanut butter production that are sprayed with herbicides. What happens to the lovely little solanums which are growing under the trees? It's not very good for them. So lots of new things have been discovered and described. But there are also quite a lot of important discoveries waiting in herbaria. Beber and et al., led by Robert Scotland, published a really nice paper in which they showed that most new discoveries come from herbaria. Many, many new discoveries are not from the field, they're from herbaria. And Chris Martin showed this really clearly by having those specimens in the herbarium in the Northern Territory, which are identified as something weird and have been sitting in the herbarium until Chris and his team go back out and look for them and figure out, yeah, that is something a bit weird. And I like this quote, which came from a paper written by Robbie Hart, again, a while ago now, 6 years ago now, that in this era of rapid environmental change, these collections that we have are increasingly important for understanding this change and also for supporting conservation. So as a result of that, we need to strengthen specimen collection, but also specimen curation, which means looking after those collections. And more importantly, even than those two, is data availability. Because unless the data from these collections are actually available, they're dark and obscure and no one can get at them. Which means that the efforts to pull the data from herbaria, from the herbarium sheets, and make those publicly available are very, very important now. And this has been a really important aspect of the last decade of work in botany in particular, in biodiversity in general, but botany in particular. And a lot of this happens in big herbaria, but actually the little herbaria are really, really, really important. Because we found in looking at Solanum in Peru with here's Maria, this is Maria Baden, Tina, Paul Gonzalez, and several other people, that looking at the distribution of Solanum in Peru, that our knowledge of the distribution of organisms in an area is actually hugely impacted by those collections in local herbaria. And Peter Moonlight recently did a very nice series of tweets about evening up access to collections, in that the local collections in country are really, really important for understanding the distribution of things locally. But we need to also, from the big herbaria in places like Europe and the United States, share our collections that enable people to make those comparisons, to make that local knowledge applicable in a global context. So going both ways, all herbaria are really important to digitize. And digitizing can be as simple as putting things in a database and making it available openly. So databasing has been a really important part of our monographic effort in Solanum, and I'll show you why that's been so important in a little while. So when we did this talk last year, I looked at those challenges I had from 2005 and thought about them again in the light of the experience that we've had over the last 15 years in monographing Solanum. And I looked at those and I thought, well, you know, a lot of these are really no longer challenges. What these represent is opportunities for botanical monography or basic botanical work to actually become more relevant and more interesting, and also for it to contribute in a much more rounded and fuller way. So time, access, and institutional commitment, yep, those are still challenges. Yep, access is still a challenge. Institutions, you know, how many of us have our institutions not saying us, come on, where's the next grant? Where's the next money? Where's the next this? Where's the next that? We need you to do this. We need you to do that. So commitment from institutions, access, and time actually are still challenges. But for me, collective working, credit for effort, common goals, specimens, and mission creep actually have become huge opportunities through the work, and we've seen that through our work in Solanum. So I'll take those first ones first, collective working, credit for effort, and common goals. And one of the great things about working in Solanum and working in Solanaceae is that this is an economically important group where a lot of people are very interested in both the species that they're interested in, like the potato or the tomato or the aubergine or Solanum torvum or gilo, but they are also interested in understanding the broader context in which those species sit. So as part of that original PBI project, one of our challenges was to link our wild species datasets to the emerging genomic resources. And that emerging genomic resource in 2004 was the tomato genome. And so as part of our PBI project, what we did is we became much closer to the genomics community and began to participate more in the genomics community in a way in which we weren't talking just to people who understood what we said or what we were about, but we were trying to talk to people who were perhaps not as conversant with the kinds of things that people like taxonomists and phylogeneticists do. And that was a challenge at first. I remember going to my first plant and animal genome meeting in San Diego, which is like this giant meeting. And I had a notebook, I had my little notebook, and I had a column down the side, and I wrote down all the words that I didn't know what they meant. And there were loads and loads and loads of them. And over the years, I kind of got to understand what these words meant. But I like to hope that the people in the genomics community have also understood some of the words that we use, like synapomorphy, like monophyletic group, like sister taxon, like the kinds of things that we do all the time, and to understand about the fact that species, a taxon name, a species, is a hypothesis about distribution of variation in nature. So it's been a really exciting journey working with the genomics community. At least for me personally it has, but I think it has for our community as well, because looking out at other groups of angiosperms, nowhere can you really see a genomics and biodiversity community as united as they are in our family Solanaceae. And I think that requires effort, and it's also really, really fun. So this means that we see these wild relatives that many of the people working on crop improvement are now seeing these wild relatives not just as individual isolated accessions or plants, but this larger network means that we look at these wild relatives in a context. We look at them in a context of what they're like and what they're doing with their lives where they normally grow. But also we, as people who look at plants in their normal lives the whole time, can also look at some of the genomic consequences that are coming from the in-depth study from the genomics community. So this has fed in from both ways. And so I think that collective working has not only worked for just a group working on monographing Solanum together, but it's also worked to reach out to other communities who are doing a completely different kind of work, who now, I think, appreciate the kinds of work that taxonomists and phylogeneticists do much more. And being part of papers like tomato genome papers or potato genome papers, where, a lot of it is pretty difficult to understand, that's been a real journey, and it's always good to learn something new. So moving on to specimens. This is the world according to Solanum. This is the solanaceous world, and this again is from last year, and I've been doing lots of georeferencing recently, as Tina will tell you. But so this probably looks a bit different now, and there are holes there where we haven't seen specimens. So these are verified collections of Solanum for which we have actually verified the identity of the particular collection. Now that verification is actually really important, and I'll come on to that a little bit later. Having the database that underpinned all of Solanaceae source and our work on Solanum monography actually has been essential to visualizing Solanum and Solanaceae, increasingly Solanaceae, in this way, to looking at where things are collected, where the holes are, where the areas where we need to collect, where the areas where we need more information are. And that's a really important way to make our work much more efficient. But it also, having that kind of database allows us to share this information with a broad set of other communities. And we currently share information with GBIF, but also with the World Flora Online, and increasingly we will be doing this through DISCO, which is the Dispersed Systematics Collections Initiative, which is happening in Europe right now, which is trying to unify all of the European natural history collections into one super research infrastructure. Because if we think about what herbaria represent to us as botanists, but natural history collections to all people working in them, is what they are, is they're an infrastructure. They're a giant research infrastructure in the same way that CERN is an infrastructure, or the Large Hadron Collider is an infrastructure which supports that research into the world of tiny things we cannot see. Herbaria and natural history collections actually support our work into understanding what the world is like. So seeing them as an infrastructure is a really important step forward, I think, for collections in general. But there are a lot of specimens. There are a lot of specimens out there, and decisions have to be made about every single specimen. And it doesn't actually happen any faster than it used to, necessarily. So looking at a specimen, deciding if it's one species, species A or species B, is a decision that has to be made. And if there are more specimens, there are more decisions that have to be made. So speeding up taxonomy and huge numbers of specimens actually are slightly antithetical to one another. That doesn't mean we shouldn't have more specimens, but we have to realize that we need to sell that message that actually more specimens means more decisions, which means more work. It's not that we don't want to do it. It's just that there's a time commitment in there as well. So I don't think there can be too much evidence. I think that having lots and lots of specimens is absolutely great, and georeferencing lots of them and figuring out where they're from, even if they're from the same place, even if they're 2 kilometers down the road, can be really important for later things, which I'll show you in a minute. So the last of those challenges, which I think has turned into an opportunity, is mission creep. Now, mission creep is that thing where you as a taxonomist are investigating a particular group and you come across something which is really, really difficult, and you decide that you're going to spend the next 25 years investigating a species pair and figuring out what's happening with them. Now, if the goal is to do a monograph of all of Solanum, spending the next 25 years figuring out one thing is not going to get you very far in getting that overall goal. So it's a question of goals. Now, that mission creep we saw it as a challenge, but actually mission creep can be a real opportunity for other people to contribute to the overall global effort to look at diversity in a big group like Solanum, which a single person or even a group of people can't really tackle on their own. But also that mission creep can extend out to other things. It means that as specialists or as monographers of a particular group, we can contribute to broader initiatives which allow us to look at large-scale global patterns with taxonomically verified and very, very accurate data sets. So the fact that there are all these specimens, they are georeferenced, they're actually identified by people who know what they're talking about, means that we can do projects like this one, which is led by Domingos Cardoso in Salvador and Bahia in Brazil, and Tina, with a cast of thousands, to look at plant species diversity in the Amazon rainforest. And in this way, lots and lots of people like ourselves got together and pulled together a data set which was verified to habit, to where things occurred, whether it was Amazonian, to have a taxonomically verified species list. Now, a species list is, of course, not an end in itself. A species list is the beginning of all kinds of things like effective conservation, effective exploration. So having that ability to taxonomically verify things is really, really important. And that's where monography becomes important, is because the global view of a group actually allows you to do a local view in a much more focused and much more realistic and less changeable kind of way. So I've participated in another project, which is to do the same thing for the island of New Guinea, which has also been an amazing journey and an extraordinary experience. So mission creep is good, but mission creep in collaboration is good. So using all of this, pulling all of this monographic stuff together, also allows us to do new and really different things with what we've done. And I'm just going to talk about this a little bit because I gave a talk about this, which is on YouTube, which I gave as my presidential lecture at the Linnaean Society about, end of May, so a while ago. And this is the PhD work of Susie Echeverria Londoño, who's now working at Imperial College. And what she decided to do is she wanted to take the big phylogeny that Tina had put together in 2013, and to look at and to do a diversification analysis of Solanum. Now that works fine if you have even sampling of all of the clades within a big genus like Solanum. Because what we thought was that, you know, most Solanum species are in the neotropics, and you can see that here. This green is all the neotropics. So most Solanum species are in the neotropics. So my expectation from this work was that the neotropics would be diversifying faster than anywhere else in Solanum. But first we had to think about how oversampling in particular clades might influence such an analysis. And you can imagine that with every single potato species being sequenced and in the phylogeny, but only 1% of the Geminata clade, which are a very rare set of forest treelets, that there would be a bias in the sampling. So what Susie did is she sat down with me and Tina, and we went through and we added to the phylogeny using a methodology called PASTIS. We added to the phylogeny all of the tips that weren't molecularly sampled, and we constrained them in various ways, which the paper is in press in the Journal of Systematics and Evolution. And so she did, you know, lots of fancy analyses, and she used the PASTIS, and she used BAM and TREPAR and various things. And what she discovered was something that completely shocked me. Is she discovered that in fact the highest rate of diversification in Solanum is happening in the Old World spiny clade, and mostly in Australia, but also to a certain extent in Africa. So that was completely contrary to the map of species diversity within Solanum, which you can see here. So here's species richness in Solanum, and the highest species richnesses, which we've always mapped and we always talk about, are in the Andes and in southeastern Brazil. You know, that's where the high species diversity in Solanum is. But the net diversification rate, which is calculated with that diversification rate also corrected for geography, is in Africa and in Australia. So the reddest areas are here in the northern part of Australia, right where Chris Martin is working. So good for you, Chris, in choosing the best place to work with Solanum. So that was a really exciting thing that we could do. We could only do that analysis because, first of all, we had a phylogeny, and second of all, we had done the hard work of trying to figure out what the species were in a broad way across the entire genus, rather than focusing just in on a little area. So this was broad coupled with deep. And I think that, you know, the numbers may be wrong. There are lots of new species and things, but I think that being able to kind of contribute to these broader scale analyses is really interesting. And one of the things I think is interesting that's come out of this is this diversification has to do with aridification. So this diversification has to do with things getting drier, and particular body types of Solanum being very successful. And in a rapidly aridifying world, this may allow us to identify some of the winners and some of the losers as we move forward through an era of what will be quite catastrophic climate change. So botanical monography, you'd never think it'd be able to help answer a question like that, but it's able to do it partly because you combine in collaboration with people doing different kinds of work. So, you know, there are mountains to climb doing botanical monography. The first of which is, why are you bothering to do that? Why don't you figure out this species concept or that species complex? But what we've tried to focus on in Solanum is understanding as much of this huge genus as possible in as even a way as possible. And by doing that, it's enabled us to reach out to other communities and to be able to do things in really different ways. So this is a very, very personal list of what a monograph should be in today's world. We think, monographs should be electronic. They need to be multimedia. They need to be fast moving. We can't afford to we can just say, here be monsters. This is an area which we don't really understand. Somebody in the future needs to do this. They need to be modular. So the days in which a Michel Félix Dunal could sit down and write an entire monograph of Solanum are long gone. They need to be incremental, put together bit by bit. they need to be inclusive. They need to include as many people as possible, as many people from as many backgrounds and as many different points of view as possible. And the one thing that they will never be is complete, because they need to be flexible and they need to be evolving. So we had this big journey, which started with PBI Solanum in 2004, and there are now more than 900 species descriptions of Solanum on the Solanaceae source website, and we're doing more all the time. There are a few big groups that are left to do, but we're definitely on the downhill slope of going over that mountain. And I sure hope that by the time I retire, or by the time I, drop dead walking in the street, that we will have got all the currently recognized species of Solanum onto that website, which means people can use them, reuse them, tell us where they're wrong, and contribute in their own ways. So both of us actually, I think, owe a huge vote of thanks to our institutions, which have supported this kind of vision of doing monography. We are very lucky to work for two institutions which have a vision which includes doing this kind of monography, but we've also been supported by a number of different funding organizations. And people always say you can't get money to do basic taxonomy, but I think we've shown that that's not necessarily true. It can be part of a lot of other things. And it actually, as part of a lot of other things, it becomes much more rewarding. But most of all, I want to thank people. We want to thank people. These are some of the many, many people that have contributed to this Solanum monographic effort over the years in all kinds of different ways. And each one of them's contribution has been as valuable as any other one's. And I think what's important about this is that you can't do this without lots of people doing it together. So I guess my vision of why do a big monograph of Solanum is you get to work with loads of amazing folks. So thank you guys very much.
Thank you, Sandy. That was amazing. Like a great story about, yes, all what you can do, like studying Solanum. And yeah. So now if there are questions, and you can unmute it, ask directly or using the chat, as you would prefer.
I'll watch the chat.
Yeah.
I had a kind of question to everybody, really. As you spoke, Sandy, I thought of the idea of, how there's GenBank. The ones who don't know GenBank, it's a place where you have to submit, if you want to publish, sequence data. And prior to publication, it becomes available within 6 months at least. It will have to be publicly available. And then there's the eFlower database for morphological data, perhaps, especially to do with flowering structures. And I wondered whether there would be a vision where similar would happen for specimen data. And I know it's not, it's a funny world because there's GBIF and there's herbarium databases, but it's not quite the same.
Mm-mm. So one of the things that we've done, and Tina knows this because and anybody who's published with me has known this, is that what I do for all of our monographs is we no longer cite any specimens in the pages of the monographs. What we do is have these giant data sets at the end, which are Excel files, which are frozen, and we publish those as data sets, which are then searchable and usable by anybody. And in a way, it's kind of like Dryad, and I'm lucky. I mean, I'm lucky in that way because my institution has a data repository. So we have a repository, and we're encouraged to do that. But I think it is super important to have all of the specimens that you've used to do a monograph available in some way or another, and actually having them available as Excel files, which are searchable, sortable, downloadable, copyable, whatever, is actually more useful than having them on a printed page of a monograph, because that is static and you can only use it in one way. Whereas, how could we, and another thing that might be a challenge is how can we use specimen data in different ways and the taxonomic data in different ways? Because we're used to kind of using it in one way, aren't we? But actually the challenge is to figure out new ways to use it and ways in which we can mine those data as well. And the reason Solanaceae source is so oddly structured, if people have gone into the descriptions, is in the olden days being 2005, when we first constructed that website, it was very difficult. Web searches were not as sophisticated as they are now, not as easy as they are now. And the idea was to parse the Solanum body into different parts in order to facilitate searches of particular parts. So if you wanted to go through and find all of the taxa which had orange fruits, you could search through and then collate all of those taxa that had orange fruits. And of course, Rebecca did it in a much more nuanced and sophisticated way, but that was kind of the idea behind having it done in that way. So a lot of the way we've done it on the website is historical. And I'm involved with the Scratchpad teams now. People who use the website will be pleased to hear in turning it into something a little bit more useful that works more often than it did in the past.
Yeah, Mario has a question.
Yeah, sorry.
Hi, Mario.
Sandy, that was a wonderful talk. I really, really enjoyed it. And I was very interested about the work that you discussed by Echeverria and Londoño. And if I understood correctly, the hypothesis for the increased diversification in Africa and Australia is aridity, drying of the environment.
Well, it's so what's interesting about those taxa. So in the paper, there's another map of Australia. So there's a closer map of Australia. And I didn't show any of those things because I gave that talk, and I've given that talk already, so that's boring. But so there's a map of Australia, and you look, and what's really cool about Solanum is, and I didn't say it, but I could go back. But is that Australia is like a controlled experiment in Solanum diversification through time. So there is a lineage, an exclusively Australian lineage of Solanum, which is one of the first branching lineages within the genus, which is Archisolanum. So that is Solanum laciniatum and aviculare and those bods. And they're all down in the moist part of Australia in the south, and there's one species in New Guinea. And essentially they are older. They're much older because they're one of those first branching lineages. And according to the dating that is in the tree that Tina and I did in 2013, but that's all going to be changed now, but I think it's not going to substantially change enough to affect the results completely, is that the long distance dispersal, or the dispersal event, whether long distance or across Antarctica or whatever, that led to Solanum going to the Old World, and then their explosion coincides with the Miocene aridification of the interior of Australia and of the western part of Africa. So that means, and the interesting thing about that as well is that the sister group of all of the Old World spiny Solanums. So one of the cool things about Old World spiny Solanums, which I could talk about for hours, of course, because they're very cool, is that they're a monophyletic group. So you think, wait a minute, there's all these spiny Solanums, there's loads of different lineages in the New World, but the Old World ones are all this big monophyletic ka-chung group, which is essentially defined by being in the Old World. And their sister group is the Elagnifolium group, which are arid-adapted rhizomatous shrubs that occur in Mexico, in the dry, arid transvolcanic belt, and in relatively arid Argentina. So in a way, what Susie always says is that those plants, they were in the right place at the right time with the right traits to just go ka-whammy and just, really, really explode. And the thing is that Manuel has just said that happened in eucalyptus. And actually, it's a real pattern in Australia. It's a real pattern in Australia that there are these arid adapted groups which have exploded in this same kind of time frame. And it's super interesting. And I think that one interesting thing to do would be to pull all this stuff together and actually do a meta-analysis of all of these different groups, because there are two different kinds of groups in Australia. There's the ones that are the arid ones and have this pattern, and then there's other ones which are more mesic, and more the better and better understanding of biogeographic barriers, especially across the northern part of Australia, is increasing our knowledge of that. And so the work that Chris and his group were doing on understanding the phylogeny and the relationships of a lot of those taxa in Australia is going to be really important to understanding the dynamics of how that pattern that Susie uncovered in a kind of very rough and ready way, how that's manifested on the ground with current diversity.
Yeah, very, very interesting. Thank you.
Yeah, so high synonymy rate in Solanum. So I'll tell you, it depends. So Ellen's asked, she was wondering about the high synonymy rate in Solanum. Well, it's because, partly, it's because we have the potato, which has 601 validly published synonyms. and we have the tomato, which has a lot of validly published synonyms, and we have the aubergine or eggplant, which also, as Xavier will tell you, has quite a lot of validly published synonyms. So it's partly bedeviled by cultivated plants, and it's partly because a lot of people, particularly in potatoes, actually, not so much in tomatoes, but potatoes more, is taxonomists in the 1940s were naming local varieties as taxa. So they were naming them using the botanical code instead of thinking about them as cultivated plants. So that means the literature has been, one might say, cluttered up with a lot of names which are validly published under the botanical code, that actually people now would probably describe them using the code of nomenclature for cultivated plants, which is dependent upon the botanical code, but is different. So it has a lot to do with those cultivated plants. But there's other taxa like Solanum americanum, which is a globally distributed little weed, which also has a gazillion synonyms. So in the most recent monograph with Tina and Gloria Barbosa, and we are monographing bit by bit the morelloid clade, which has in total about 80 to 90 species. And in the most recent installment of that, we did North America, Central America, and the Caribbean. And in that, there were 19 species, and 3 of those species had more than 100 synonyms each. So not fun. And working all that out and European plants as well have a lot of synonyms. So there are a few Solanaceae, there are a few Solanums in Europe, but they've got loads and loads of names.
Thank you very much. Yeah, and there is a previous question, and we're having several ones of Stacy actually, like because she mentioned about that. You said a good deal about all the pieces that are needed. But the thing that she's worried about is training, actually. How can we ensure that we are training the next generation in taxonomy? That's her question.
That's a really important question, and that's something that the taxonomic community has been struggling with. And I think it's because we've been, to a certain extent, struggling with it in slightly the wrong way. So this is not a popular view. So what I'm just going to tell you is not a popular view. And I get shouted at a lot by people for having this view, but it's always good to get shouted at because it means somebody's thinking about what you're saying. I think actually taxonomy is quite easy. Taxonomy is just comparative biology. Taxonomy is about looking at things and telling whether they're the same or different. I used to tease my kids to say that basically what I do every day is which of these things is not like another, which is that thing on Sesame Street, that you have 5 things and one of them's different. But essentially, if we train people to think comparatively, then we've trained the next generation of taxonomists. To one extent, we don't need to train people who are just like us. I would be sad if everybody who'd gone through my clutches throughout the years was just like me. I'm really happy that they've taken what I've been able to give them and taken it to new heights and have done it differently. So I think it's a mistake for taxonomists to think that we have to train people just like us. But I think it's that recognition that taxonomy is comparative biology, and it's the organismal comparative biology which we need to be sure to retain. And that I think the pendulum is slightly swinging back, but we need to be very loud and vociferous. And I think show people how that can contribute to all kinds of solutions of what people see as societal and global problems. Because you'd never think a monograph of Solanum could actually contribute to conservation in the Amazon necessarily, but it certainly will because of the work we've done collaboratively with other people. So I guess the training is really important, and I think that Stacy is right. She said, what are the best ways of training and supporting the next generation? I'd say comparative biology, but also societies will have a great role. And I think that doing, because I've become, slightly by accident, I've become a quote unquote expert in nomenclature, which is like not the most popular thing to be an expert in. But what we've done to do this is IAPT, the International Association for Plant Taxonomy, has sponsored nomenclature courses at many meetings all over the world. We do them at the Botanical meetings in the United States. We're doing one, we were scheduled to do one in September of this year. I think it was September. No, it was July. It was now. We were supposed to be in South Africa right now, teaching nomenclature in South Africa. So the IAPT has done that, and I think it's really important that those of us who are interested in nomenclature and are willing to take the time to do it actually, devote the time to go before and after those meetings and actually give courses that are not, everything you wanted to know about nomenclature, but kind of what's the most important stuff to know. Because there's only a few things in nomenclature which are actually really, really important to people. They're really important for naming things and for understanding synonymy. There's a few things that are important, and the rest you can look up in a book. So I think societies will have a really important role. So somebody else asked.
I just wanted to add that I feel there is a lack of opportunities when you come up. So it's really nice to see projects that are openly acknowledging that some taxonomy has to be done. So when you make proposals for funding, for example, not to put it under the carpet, not to say we're going to collect 1,000 specimens and it costs nothing, but actually say we're going to collect 1,000 specimens and processing cost X amount, or that in a PhD there's a little bit taxonomy, kind of like Mario Vallejo Marin often has students who take into account that maybe it's not fixed in stone. That maybe if I bump into a problem that the species isn't monophyletic, at least I test it. And that secondly, if it isn't monophyletic, I reach out to a taxonomist and learn a little bit. That if that kind of open-mindedness would be coming from different fields, that if I reach beyond my field in my science as a taxonomist, I often bump into, oops, I don't understand reproductive biology. I reach out to somebody like Mario and say, hey, could you help me? And I guess we can all do that in our bits, because without that, the student grows up thinking everything's fixed. We don't need to test the hypothesis, is this species monophyletic? We do phylogeography of species X without ever questioning, is it a single species? I mean,
so I think collaboration.
They need to be embedded in everywhere.
I mean, collaboration works two ways, though. So it requires taxonomists to also have an open mind.
Yeah.
It's not just the other people who have to have an open mind. And I think I think it's super important for us to kind of say, okay, how can I do this in a way? And it's challenging. I tell you, it's pretty challenging. It can be quite challenging giving talks about species concepts to genomics meetings. It can be pretty challenging. Quite fun though. It's quite fun. When the first time I ever convinced anybody that something that phylogeny was worth looking at, in other than a way of saying tomatoes are X, was amazing, because it's like scales drop from people's eyes, and it's sort of like, wow, I can think about this in a different way, in a different framework. And I think the thing about taxonomy and phylogeny is that it is the framework, and it's our framework, and we kind of do all of our work in that framework. But it's very, very difficult to understand if you're not in it. So having in a way, just kind of being open to learning from other people about how difficult it is sometimes makes you do things differently in the future. So I once got asked by somebody whether giving lots of public talks in the Natural History Museum made me do my science in a different way. And I was with somebody else, and they said, of course not, we would never do our science in a different way. And actually, I think that's the wrong answer, because it does make you do your science in a different way. It makes you think about your science in a different way. It makes you think about how you're asking questions. That interactive thing is so important, because we don't have all the right answers. I've been doing things in a particular way. I know there are people who have worked with me who are sitting here in the audience, and you will recognize yourselves, is that I've done things in a particular way. And somebody said, why are you doing it that way? And I thought, I don't know. I've just always done it this way. And then they had a much better way of doing it. And it was great, because this is fantastic, because I just became like uber efficient suddenly, which was wonderful.
Okay, there is another question also from Federico Roda, that he asked about if you think that violence in tropical countries like Colombia could have caused an underestimation of diversity.
Yeah, I think violence per se does not necessarily cause an underestimation of diversity. But the fact that people can't go looking for things in areas or can't contribute to more global efforts, yes, does cause underestimations of diversity. And for example, if in countries like Colombia, you have the inability of institutions to digitize collections or to make those collections available, or to invite specialists to come in and teach and you know, all that kind of stuff, then that does contribute to underestimation, can contribute to underestimation of diversity. So yeah, I think it is important that that we open things up, that things are open, and one of the things that the Royal Botanic Gardens queue is they have a very big program in Colombia, and they're collecting in a lot of the areas that were previously marred by violence, and that's going to be really important to understanding things. But it's also important for Colombian scientists to be able to access the historical material that comes from Colombia, which is held in European herbaria. So that's one of the important things about these old crusty herbaria like Paris, like London, like Madrid. Actually digitizing all of that material is to make it available, because that's also part of understanding the diversity of a country.
There's a great question about Russia being an empty country in the specimen records in our map, and it is because they don't have solanums. It is, I would say, simply because their language is in different alphabets. We can't. How do you database and how do you access it?
No, that's not the reason. No, that's not the reason. There are very few solanums in Russia. Because I've spent with Maria Vorontsova, we went to St. Petersburg twice, and I went through the whole of the Russian collection and databased most of this. There are about 4 species of solanum in Russia. So
I think if you would have the Solanum americanum and nigrum, because they're so weedy, we'd have dots. It's just that there's very little.
Yeah, maybe.
I'm guessing.
But
most of the few weedy species would be more.
Much of Russia is above the area in which those species grow. Because when you look at the projection, of the map, it looks as though that part of Russia is the same latitude as everything else, but it's not. It's way farther north. So it's farther north. So there are very few. So there's about 3 species of solanum in Russia, and it's huge. And when I did the monograph of the Dulcamara group, I was really confused because there was some stuff in northern Japan which I didn't really understand. And then there was all this stuff in Europe and in North America. So Solanum dulcamara is this thing that goes over the top of the northern bit of the earth, right? And so I went to St. Petersburg and basically went through, and my son, who speaks Russian, I paid his ticket, and I made him work for me for 6 hours a day in the herbarium, transliterating the labels. And then he got to go and explore St. Petersburg for the other 4 hours a day that I worked in the herbarium. So we databased all of the Solanum dulcamara across all of that. And what was fascinating about that is there are collections from Siberia in the 1930s. So what were those people doing? Canada too is empty, but Canada also has very, very few species. It's really, really a Southern Hemisphere group, Solanum.
Okay, I think, yes, we're running out of the time, but yes, it was a great questions and a great discussion of all the Solanum.
Thank you guys very much. Thank you for
coming. Thank you very much, Sandy, and also Tina, for this great talk. And thank you everyone for coming and yes, for sharing and listening to these talks.
And can I just say, Rocio, thank you to Rocio and Chelsea and Andres for turning the Phyceloid seminars into the Solanaceae seminars online, because I think that's a great thing that you 3 have done. And it's a super important thing for us to do going forward, because all of these online talks are much more inclusive and we can all collaborate in a much more even way than we could before. So congratulations to you 3 for getting it together.
Thank you. Well, we expect to see you again next Friday with a talk of the Comstead about the historical perspective on classification and molecular phylogeny in Solanaceae. So that's great. And it's great that, if you want to turn on your videos, it's great to see you all.
There's a very small person. There's a very small person there who's now going to be a solanologa.